So we have been working on a little bit of our rose black spot disease resistance, but mostly on developing markers that are associated with the loci that we have already identified. And the people behind all this work, I just wanted to present them before we started, are here at the University of Minnesota, myself and Seth, that is joining our team and will be soon taking over the Rose Black Spot project here at the University of Minnesota. We work with our undergrad, Nina, that is the one that takes care of the plants, and Emily, that works on the population Northern Blush by G168-2. And over at River Falls, we have a David that works with several undergrads, so Jake Petronak and Leah and Eliana over the summer to do an immense work on genotyping and phenotyping for rose black spot disease, but we'll talk about it in more detail. So the different areas of focus of our lab group is developing segregating populations for loci mapping, and then lately it has been to work on developing the markers of the associated with the loci that we have already been publishing. So in the greenhouse, we have David that has been working on finishing up the development of the population that is a cross between Northern Blush and a selection that has Applejack in its background, as well as Orange Honey. And I can't remember the name of the other ones. But yeah, the most important is that it has Applejack in its background. And so then we have also, David has been developing an immense rose collection that is used for RBS screening and also marker validation. Here at the University of Minnesota, we are developing the markers that are then tested by David over at River Falls. And with Seth, we have been lately working on developing an RNA-seq experiment to find map RDR4. So last time that we met, we were finishing up the validation of RDR3 and starting the validation of RDR6, the markers associated with those loci. Actually now we just started the marker development of our associated with RDR5 from the Rambling Red. And soon, hopefully in 2025, beginning of 2025, we'll be starting the market development for RDR4. So I'm going to just go over the different activities that we've been working on since the last time we met. So first is the creation of that extensive rose collection that is used for rose black spot screening and marker validation of RDR4, RDR3, 4, 5, and 6. So David has gathered about 400 plants in total that depict 267 unique roses. These roses have been selected because they are on RDR carrying cultivar pedigree or also close relatives, but also because of their repetitive rose rosette disease resistance, like some of the Ag Canada cultivars, the Laughter, Base Thornless, among many others. And so all this work has been done there by David, and thanks to the Rose Society, the American Rose Society funding, he was able to gather all these roses and get some help from undergrad students there. So these roses have been, most of them have been genotyped using 6 isolates that represent race 3, 8, 9, 11, 12, and 13. And they have been phenotyped for qualitative resistance. So, so far, 93% of the rose isolate combinations have been completed by David and his 2 to 3 students there. So here you can see a picture of the immense work that they're pulling out over there. So for the first results that we can get, we have 133 roses that are actually susceptible to all the 6 races that were tested. So that's about 50% of the roses that have been gathered for this collection. And then out of those results, we can also see that there are 33 roses that have RDR known resistant patterns. And so, for example, there are 10 roses that have a similar resistant pattern than George Vancouver that carries RDR3, 10 that may have RDR4, 8 that may have RDR5, and then 5 that may have 5 that may have RDR6. So this is really interesting findings that David has been able to get there. So then we have been working on the development of a marker tightly linked to RDR3 and its validation. So last time we met, we had a list of 14 SNPs that could be used as markers, and so we convert them into Cas markers, and we tested all the cultivars that were confirmed to have RDR3 with the SSR markers in a previous publication by Jason. And now we have narrowed down the region to a region of three markers that are associated with RDR3, and we are validating those 3 markers on the extended collection that we talked earlier. And so far, I believe there are 208 cultivars that have been phenotyped and tested, and they are descendants and ancestors or close relatives of George Vancouver. So here we have an example of one run for Yeoman Rose SNP 16, and you can see here the susceptible homozygous roses, and then the resistant heterozygote roses, and then some other roses here that seem to be homozygous for the other allele. So reducing that region allowed us to go from 2 megabases to 151 base pair, and we went down to 12 candidate genes with 2 NBSRR genes and 2 ROP interactive types that are also known to interact with NBSRR. So this is a big win, and I think these 3 markers will be available for breeders to screen their populations if they need or if they need. And then one final question that we would like to answer with these 3 markers are which wild species did RDR3 originated from? And so that's David's work right now, trying to figure out where that RDR3 came from. Then for the marker development for RDR5, we are planning on doing a fine mapping of this region to find markers that are tightly linked to the causal gene. And we're using a different approach than what we did for RDR3. Since this time we're going to be using some GBTS, so genotype by target sequencing, in order to saturate that region, and then you use the collection that we mentioned earlier to do some sort of association mapping to increase the recombinations and narrow down the RDR5 region. And so this is what is happening right now with the collaboration of 3CR, and they are developing the props for the target sequencing, which will be then we'll be then genotyping 4 plates, of worth of genotypes with the population and some of the genotypes from the collection. Then we're going to select some of the SNPs, convert them, and then 3CR is going to validate those on more roses later on. So and that's thanks to the Glockner Foundation grant that we obtained this summer, that we'll be able to do that. Then for RDR6, we already had some SNPs from BSA-seq that were found to be associated with the resistance. And so we had 337 SNPs in that region that were in simplex configuration and 74 SNPs from the integrated map. So we selected about 64 of them that had the highest lot score for the BSA, and from the 74 will be starting with the peaks, so markers that were in the regions where the peaks were detected, and then moving outward to test those and find about 3 or 4 markers that are well linked to RDR6. So that's the work that will be done, has already started a little bit at River Falls by David. And so then what's next? We will be working on RDR4 marker development, but this time it will be a project that has a lot of components. So the objective is to find map that region and find a causal, or at least a list of causal genes later on. And so the project has 4 components. The first one is developing a genome for bright eyes, which will be the first genome of a resistant genotype that carries a resistance that gives a complete resistance to rose black spot. And then the second step would be to perform a fine mapping similar to what we are doing for the rambling red population, so RDR5. And then develop those markers, and on the side and associated with it, we will be doing some RNA-seq for transcriptomic study. And so that's with the pilot sequencing program here at the University of Minnesota, and maybe with the HRI grant. Otherwise, we have other funds that we can use for this project. And so, since last time we met, we have been working on doing some preliminary tests for the RNA-seq study. And so first, we needed to know which races we were going to use as a comparison. So we will be using only bright eyes, leaves of bright eyes, and then we selected race 12, which is the one that infects bright eyes, the only one that infects bright eyes. And then we selected race 10 that is resistant. Bright eyes is resistant to race 10, and so it yields an incompatible interaction. And so in that way, we will be able to compare better between bright eyes that is infected and that is no longer resistant, and bright eyes that is resistant and doesn't get infected. So here you have orange honey, the little leaves here are orange honey as a susceptible control, just to check that the inoculation went well. And we tested 10, 11, and 13, but we selected 10 because it grows on orange honey at a similar rate than race 12, whereas 11 and 13 take a bit longer, which is not ideal for the RNA-seq study. Then the second question is, which leaves should we use, knowing that we need a lot of leaves for all the time points that we want to do. And so we wanted to have a range of leaves that we could sample from. And so we defined stages from 1 to 6. So you can see here. And we didn't go further than stage 6 because from previous experiments, we noticed that diplcarpin takes much longer to grow on them. And so this experiment showed us that we should actually stop at leaves from stage 4. Since after that, the inoculation takes much longer. So this is at 7 dpi. All these leaves ended up completely susceptible and infected by 14 days, but stage 5 and 6 do take longer to get infected. And so that's why we needed leaves that get infected at a similar rate. So we can sample from leaves from stage 1 to stage 4. And so this is the experimental design that we would like to do with SET. So like I said, for the incompatible interaction, we use race 12 on bright eyes. For the incompatible interaction, we use race 10 on bright eyes. And then we will have two controls, a mock inoculation and one where it's just sampled from the leaf directly to look at constitutive expression of, for example, NDSR or PR type of genes. And so the different comparisons that we will be able to make will answer different questions. And then we want to do 7 time points. Mostly associated with 3 dpi, between 0 and 3 dpi, because that's when the resistance actually happens. From previous studies, we've noticed that. And so this is the overview of the projects and the fundings that have allowed us to do that. So RDR3 is the American Rose Society grant that allows us to finish the phenotyping and the testing of the markers. The Glockner grant is the one that is helping us do the fine mapping and the marker development for RDR5. The RBS also helped us for the phenotyping and some of the genotyping of the CASP for RDR6. And then Bright Eyes will be funded by the UNGC for some of the RNA-seq work, and then maybe the HRI grant or some funds from our lab directly. So thank you so much for your attention and for all the people that have been helping us in this project.
Thank you, Diana. Any questions for Diana? But we have to be quick since we're running out of time. If nobody has a question, I just have a quick one for maybe Diana or David. Does Miracle on the Hudson have RDR3?
You want to answer, David?
Yes.
Go ahead.
So the answer was yes?
Yes. How did you know?
The reason I ask is I think we are detecting the activity of RDR3 in the Miracle on the Hudson by Morton Blush and Miracle on the Hudson by Morton Fargo populations here.
Okay, good.
And next thing is I'll bug you about how I want to map that gene more accurately in my population so that it kind of comes together properly. Okay.